organizing committee for inviting me today and giving me the opportunity to talk about some preclinical studies, particularly some of those we've been conducting more recently at the University of Colorado, the Anschutz Medical Campus, just down the road, where we're basically focused on developing new technologies which I hope some of the neurosurgeons and neurologists here, many of you will use in the future to promote repair of spinal cord injury and actually probably a variety of other neurological disorders. So I was asked to focus on some stem cell technologies that we're working on in the lab, and I'll be talking today about means of making the right astrocytes for central nervous system repair. So I'm going to start off by giving you a bit of basic background about why we think axons fail to regenerate in the adult central nervous system of mammals. Some of the basic theory, and then I'm going to talk about some old experiments that I conducted that really helped us focus on astrocytes, particularly what types of astrocytes we want to make from stem cells for repair, and then talk about some preclinical studies where we've been looking at the ability of stem cells to make the right kinds of astrocytes to give us axon regeneration across injuries, function repair, and avoid some nasty side effects you might have if you use stem cells in inappropriate ways for repair of spinal cord injury. Okay. So a bit of background. Spinal cord injury. There was a survey by the Christopher and Dana Reeve Foundation in 2008 that showed there are about 1.26 million people in the US alone suffering from disabilities associated with spinal cord injury. It's still the majority male compared to females that are getting these injuries, and the average age is 33 years of age now at that time of injury. We have an aging chronic SCI community, with the average age now between 40 and 59 years of age. Only 18% of spinal cord injuries result from sports and recreation injuries. We imagine there's somebody on skiing or on a snowboard hurtling down the mountains here, and sure enough that they're at risk of getting a spinal cord injury. But it turns out the leading causes of spinal cord injury are motor vehicle accidents and falls in the elderly. And at the moment, despite there being a few ongoing clinical trials, there are no FDA-approved sort of effective therapies, certainly in terms of interventions, either drug or cell-based therapies for spinal cord injury. So as we have a mixed audience here in terms of expertise in central nervous system and some of the theory of nervous system repair and regeneration, I'm going to give a quick sort of overview, some basic overview, and then we're going to get more complex in terms of talking about astrocytes. So, the brain and spinal cord. We have open motor neurons in layer 5 cerebral cortex that have axons that project all the way down the spinal cord and known as supraspinal pathways, and a lot of involved in motor control. And so I'd like to actually make this point right now, actually, that when you enter the spinal cord, you're actually directly injuring these neurons in the brain because they don't like having their axons cut. A lot of them degenerate, and a lot of changes, and some die. All the changes in the brain after spinal cord injury, and so we really shouldn't consider brain and spinal cord injuries being somehow in any way separate. And then there are neurons which lie outside the central nervous system. In this case, we could consider dorsal ganglion neurons that send their axons into the spinal cord from peripheral nervous system to central and project to the brainstem and to the brain in general. And so the idea being then that the higher up in the spinal cord you have an injury, the worse off you are, because you've now disconnected inputs from brain to spinal cord if you have a cervical spinal cord injury, and you've lost all that sensory input to the brain. And so obviously people like Christopher Reeve was severely disabled when they had an injury at sort of C1, C2 level. Okay. So in cross-section, this is a schematic of human spinal cord, and you can see the classic sort of butterfly shape of grey matter in the centre of the cord, and we get surrounded by white matter pathways where these ascending and descending axons reside. And so there's obviously nice topographic mapping of different motor and sensory pathways in the spinal cord. And after spinal cord injury, as many of the neurosurgeons know here, we have a process known as hemorrhagic necrosis. There's bruising of the cord, contusion injury, and we have bleeding from the inside out. And my understanding is that part of that process is that grey matter has 5 times the density of blood vessels, white matter, and so it's more vulnerable to contusion injuries where you then have that bleeding in the centre of the cord. And so my understanding as well is that there is a particularly thoracic spinal cord injury patients walking around who maybe fell off a ladder and had a relatively minor injury where the bleeding and the hemorrhagic necrosis in the centre of the cord wasn't that large. And so the idea being that the injury didn't impinge upon surrounding white matter, and there wasn't therefore that much of a loss of function. But if you have a larger injury, and now that bleeding and damage impinges upon the white matter pathways, you're disrupting that flow of information up and down the spinal cord, and now you have major disabilities in patients. And as it's now been well characterized, you have a cavity forms at the injury site, of demyelination around the injury, and formation of astroglial scar tissue, usually with a fibrotic center full of macrophages at the injury site. So, over the last 100 years or so, we're trying to think, why is it that there's this failure of axons to regenerate across injury sites, in particular, within the adult mammalian central nervous system. And it's long been known that the relatively immature central nervous system, particularly embryonic mammalian central nervous system, has a far greater capacity to recover from traumatic injury and an ability for axons to grow. So in the schematic here, you can see the cell body of a neuron within the central nervous system. This is axon. And after exotomy, damage to that axon, you have a process known as Wallerian degeneration, which is named after the famous 19th century anatomist Augustus Waller. And the idea is then that the part of the axon distal to the injury, disconnected from the cell body, degenerates and dies and is lost. But in the immature central nervous system, prior to clearly defined time points in development, and this actually change takes place just after birth in mammals, postnatal day 2 to 3, for sensory axons in spinal cord. Before that, axons are able to re-establish a growth cone of the injury site, grow either through or around the injury site, and re-establish functional connections. However, in the adult central nervous system, adult neural cell body here, a lot longer axon in an adult in general, after the same sort of trauma and exotomy of the axon, once again Wallerian degeneration, but unlike the early nervous system, and see that there's a really clear cutoff point, certainly for sensory axons in spinal cord, postnatal day 2 to 3. Before that, get good growth across the injury. After that, into the adult, what you find is now axon, the tip of the cut axon fails to form a growth cone that's able to traverse the injury site, and you have what's called a dystrophic ending forming at the injury site. First, that term was first given by Santiago Ramon y Cajal, famous Nobel Prize-winning neuroscientist who was very interested in trying to promote the paracentral nervous system. So he first described these dystrophic endings. It's rather pathetic some endings that axons form, and they fail to cross the injury site. So one thing you might consider is, okay, from the embryonic nervous system to adult, what's changed in the pathways? We're thinking about trying to promote axon growth, particularly white matter, because the disruption of white matter in spinal cord injury gives you a major loss of function. So I think we're focusing quite a lot on white matter as we go through this talk. What changes have occurred during development of spinal cord pathways, white matter, that may in some way give us clues as to why axons have failed to regenerate just after birth? So this is just some studies conducted by Suzuki and Raisman at National Institute of Medical Research, Jeff Raisman's lab, where I actually studied my PhD. I was actually involved in some of these studies. And so looking at hippocampus, they were very interested in development of the fimbria and how the white matter pathways, what was the cell architecture of white matter, and how did it change during development. And so the idea is that, well, the CA3 pyramidal neurons and other pyramidal neurons in the hippocampus, they grow their axons relatively short distances, because we're talking about growing in an embryo. And of course, this is also true for spinal cord in respects. The distances the axons have to grow when they first grow in development are relatively small. And the glial tract architecture is very sort of rudimentary compared to an adult. You have radioglial processes, and you can see this coming out in green, and you have a lot of multipotent glial precursors, multipotent stem cells, which I'll be talking about later, in the more ventral aspects of the white matter. So these axons are growing, making connections, and then most of the axon growth during development is actually stretching of the axon, still in contact with its target. That's a very different type of growth from what we're asking now in the injured adult central nervous system. The tracts are far longer, and so you're asking an axon now to grow far greater distances to re-establish a connection. Very different type of biology. Also what happens is then this beautiful, I think, glial architecture builds up around the axons as they're stretching during development. And so this would be some for P0, 4 right to P60, when you're a relatively mature glial cytoarchitecture surrounding these axons in white matter. You can see in this little state that it changed this beautiful, I think beautiful, intervesicular glial rows, many contiguous cell types that are aligned with the axons. So we can see here, we're saying for myelin as well, in a semi-thin. We can see the oligodendrocytes lined up in a particular middle row, and we've done some immunohistochemistry here for astrocytes with GFAP, and you can see that they have the arrangement of processes in white matter. So if we make a schematic out of that, what we find is these rows of oligodendrocytes. You get about 8 to 10 oligos in a row, and then you have solitary astrocyte in the row. 8 to 10 oligos build microglia thrown in, solitary astrocyte. You never see 2 astrocytes next to each other in a row. And the spacing between these rows is highly regular in different white matter pathways throughout the whole of the brain and spinal cord. Every white matter pathway has this cytoarchitecture. And so in thinking about stem cell technologies for repair of spinal cord, you want to get back to that structure. That's what the optimal way of repairing that white matter pathway. And so you have oligodendrocytes and sending out processes and myelinating segments of axons that run parallel to these intervesicular glial rows. So over the last 100 years or so, there are 4 main theories that have been put forward to try and explain why we have this change in development in terms of the ability of the injured adult central nervous system versus embryo to repair. So 4 main theories. Why aren't axons regenerating? And the first is, well, okay, that there are changes within the neurons, intrinsic changes, that the idea that older neurons just have a lower capacity to mount a regenerative response. And there's a variety of genes that we know that are downregulated in the adult compared to embryonic neurons, such as integrin receptors, that are important for allowing axons to grow. So there are changes which are intrinsic to the neurons. There's also a theory that was put forward by Cahal, I would say over 100 years ago, the idea that there's a lack of trophic and substrate support. There's a downregulation of molecules in the adult central nervous system that could help with axon growth. And so that's why he coined the phrase, rather, or the description of his axons, in that they were dystrophic axons, lack of growth factors, where the reason he thought axons weren't regenerating. And there's a more recent theory put forward by Martin Schwab in Zurich, that in addition to loss of growth factors and changes in neurons, there are now molecules present in white matter associated with myelin that are actively inhibitory for axon growth. So loss of growth factors and now have accumulation during that myelination of the nervous system postnatally, where you have axons of you have accumulation of axon growth inhibitory molecules, no-go, myelin-associated glycoprotein, and OMGP, oligodendrocyte myelin glycoprotein, to name but a few. And then there's the last theory, which is one of my favorites, is that glial scar tissue is developed directly at the site of injury, right in front of those axons, where you see axon regeneration fail, that glial scar presents a combined physical and molecular barrier to axon regeneration. So when I was a postdoc in Cleveland, actually when I started when I was a graduate student in London, I started trying to design preclinical in vivo experiments in rats, live rats, that would give a clearer distinction of the relative contribution of each of these theories to overall failure of regeneration. Particularly wanted to find out how important is the scar versus the white matter pathways beyond in terms of what's stopping axons from regenerating. So the way I went about it, and this is actually was a follow-up experiment in spinal cord to experiments we originally conducted in the brain, was to use the adult rat spinal cord injury model where we could make a discrete transection injury at the C1, C2 level and basically cut axons in the dorsal columns in the rat. So we're going to get a nasty scar form at this level, and then the idea was to use a microtransplantation system I developed in London to inject, in this case, fully adult neurons from adult rats, 3-month-old rats, dorsal ganglion neurons, no added growth factors, minimal central media, and inject them directly into degenerating white matter beyond the injury, where it's full of myelin debris, and test, is it that the adult degenerating tracts are inhibitory to axon growth, or is it the scar that's the problem? So we're going to put these neurons beyond the glial scar, and now test, is it that the pathway won't support growth, and it's full of myelin debris, and these are adult neurons that we're putting in a real Frankenstein type experiment. Okay. And so, of course, what we found was something very interesting. What we're looking at is a parasagittal section of the adult spinal cord above the injury site, where we transplanted the neurons. You see them in green, they're staying with GFP, green fluorescent protein. This will be the peeled surface. We're getting good survival of the transplanted neurons, and we've injected them so gently that we're below a critical threshold of damage, that we're not getting scar formation at the interface of these grafts with the adjacent white matter. and we're getting great axon growth up and down the tract of 1 to 2 millimeters per day on these transplanted neurons in degenerating adult white matter and adult rats. And so now, Martin Schwab developed this myelin theory of inhibition of axon growth, and one of the ways he developed that theory was to take neurons, dorsal ganglion neurons, and co-culture them with myelinating cells, the oligodendrocytes. And indeed, as he saw, we also saw that in red is the oligodendrocytes, green neurons. These same neurons that you might transplant, you can put them in tissue culture with oligodendrocytes, the myelinating cells, and there's very poor neuron outgrowth. Indeed, these oligodendrocytes seem to be inhibitory for axon growth. And yet, if you transplant the same neurons into the adult central nervous system, the injured spinal cord, beyond the injury, you're getting great axon growth. So how do we reconcile these two data sets? Well, that was at this point started realizing that, well, there's a lot of astrocytes beyond the injury site. There's some, there's some that are forming scar, which I'll show you in a second, but there are a lot of adult astrocytes away from the spinal cord injury. And in these studies, we saw with electron microscopy, a lot of our growing axons were closely associated with adult astrocyte processes in adult white matter. So first became very interested in the astrocytes' ability to support growth from these experiments. And these images here, I think I'm right in saying, were the first images of adult growth cones of adult axons growing in adult white matter. And in particular, you can see here we have this beautiful streamlined growth cones indicative of rapidly growing axons, not making guidance decisions, and they're growing in that adult white matter structure. So that adult structure I told you about with the rows of glia, or neurocytes and astrocytes, in this case was supportive of axon growth. Now, you notice in this transplant, the growth is bidirectional. There are axons going back down towards the glial scar. And what happened was, when these axons encountered the glial scar, they stopped dead in the tracks, and they formed dystrophic endings, just like Hal showed us over 100 years ago, but only upon interacting with the environment of the glial scar. Because you remember, when you first make an injury, and you're looking at the endogenous axons, those axons that were already in the spinal cord, they've got endings that immediately form a dystrophic ending. So you didn't know whether it was something intrinsic to the neurons, or whether it was environmental scar, or a whole environment was inhibitory that was forming, making those axons form as dystrophic endings. And these experiments showed that, in fact, there seemed to be something about the molecular environment of the injury site that would make axons stop and change those streamlined growth cones into dystrophic endings. So in general, after trauma to the central nervous system, you have trauma and inflammation, you have severing of axons, you have loss of myelin, you have loss of neurons in glia, and you have formation of axon growth inhibitory scar tissue directly at the injury sites, which these experiments were showing. So in our lab, we're developing two spinal cord injury repair strategies. The first, which I'm going to tell you about today, is to develop stem cell technologies for making the right kinds of astrocytes to support axon growth. And in this case, we're looking at the ability to bridge injury sites. And as I say, as these experiments went on, you'll see that we have to be very careful how we use stem cell technologies when it comes to repair of the central nervous system, in particular what types of astrocytes you make. And the other technology we're developing in the lab is to desensitize neurons to the inhibitory effects of myelin and to the particular molecules in the scar. I think myelin is still inhibitory, maybe slowing axon growth, even though our experiments really put a big dent in the myelin theory. And so we're working with a molecule called decarin, which we hope to take to clinical trial in the next couple of years. But today we're going to focus on our astrocyte technologies. So if we, here's a schematic of a spinal cord injury. We've got ascending and descending axon, white matter pathways. We've got grey matter in the center of the spinal cord. And the grey matter, as I'm going to show you in a minute or two, is actually full of chondroitin sulfate proteoglycans. Some of these proteoglycans are in fact known to be potently inhibitory to axon growth, and yet they're found in the grey matter of normal spinal cord. In fact, some of them are known to be the most abundant proteins in the central nervous system. Some of these chondroitin sulfate proteoglycans, CSPGs, okay, in grey matter. Okay, so if we were able to modify the injury site, maybe either with an astrocyte bridge, as I'll show you in a minute, or we were able to suppress the expression of the inhibitory CSPGs at the injury site, because these same molecules found in grey matter are also found in the injury site, and we were able to alter that environment, the growing axons would have to then adapt to the substrate molecules that you find in the injury site, have to adapt to the injury site. And then, more importantly, they're also going to have to be able to then adapt to the white matter pathways beyond the injury, which are full of myelin and may have different substrate molecules. And so one important concept here is to try and make sure your bridge is well matched to tissue either side, so you're not asking axons to change their receptors all the time and continually adapt to environments because axons don't like doing that. And so primary axons that get across an injury, that's great, you've got them, say you've got them regenerating down a pathway, but that's not actually how the central nervous system makes the majority of its connections. It's actually side branches or collaterals from these axons that invade adjacent grey matter, and that's how the majority of connections are actually made in the spinal cord. My understanding is in fact that the cortical spinal tract, the primary axons in development, they grow down the pathway, the primary axons don't actually ever form connections to the spinal cord, it's collaterals all the way up down the spinal cord. That's how the CST makes the majority of its connections. Okay, so this is a concept we'll have to think about, we'll think about later, is plasticity, central nervous system, the plasticity, the ability to make those collaterals, as well as get axons across injuries. And of course, the majority of spinal cord injuries in humans are incomplete, anatomically incomplete injuries. And so the idea would be if you've got spared axons, can you also induce collateral formation from them as well? Okay. So there's been a lot of work done trying to, done very good work actually elucidating which molecules associated with myelin can inhibit axon growth. And we've got a list of them here. So it's Nogo 66, Martin Schwab has really pioneered the whole myelin inhibition theory, and we've got a variety of molecules associated with myelin and a variety of receptors. In fact, that's the story's now got 3 receptors, Nogo receptors 1, 2, and 3, and a variety of receptors that then, through ROC and ROA signaling, will shut down the ability of the axons to assemble a microtubule assembly in their axons to allow for axon growth. And so now we're realizing, in fact, that the same receptors, the Nogo receptors that are important for myelin-associated inhibition of axon growth, are also important in the proteoglycans signaling and how they suppress axon growth as well. So our work, the old studies have really shown that axons have a problem crossing spinal cord injuries, and particularly environments rich in chondroitin sulfate proteoglycans at the injury site. So we decided, okay, let's go in and find out what molecules are in there, and how do the levels of these inhibitory molecules, these CSPGs, change from acute to chronic time points after spinal cord injury. This is all the dog rat spinal cord injury research. So the proteoglycans, they've got names like Neurocan, Brevican, Phosphocan. Neurocan and Brevican are predominantly found in grey matter in the spinal cord. And this is actually a histological section where we've stained for Neurocan in green, and that's white matter versus grey matter. And the Neurocan is found in what is called perineuronal nets, matrix nets, dense nets of this molecule that sit around neurons. We think they're important for stabilizing synapses. That's what the Neurocan is normally doing in the central nervous system, because you can imagine we're also trying to grow axons into this environment to make connections, collaterals. And so Neurocan and Brevican are the 2 most abundant proteins in the central nervous system. I think Stephen out there said they're that abundant. And there's been a lot of interest, this is Agrocan, it looks like a bottle brush, in terms of a lot of interest in the sugar side chains, the GAG side chains, glycosaminoglycan side chains. And so we know that these sugar side chains can also signal inhibition of axon growth. So this is one of the elements of these proteoglycans which inhibit axon growth. We also know the core proteins of these neurons can also have direct signaling through receptors on axons and prevent axon growth. Okay. And there are a variety of other molecules that have been described in the SCA. Semaphorins in particular are thought to be bound to the proteoglycans and also inhibit axon growth. And our labs we've actually regulated semaphorins in spinal cord injury with Decarin, but that's another story. Okay, multiple inhibiting molecules in the SCA. So we wanted to look at how these molecules change from 24 hours to 6 months, and you'll see in a second why this is important in considering what types of astrocytes you make from stem cells. So these are confocal images, parasagittal sections. We've made a scissor cut to the spinal cord, and we're looking at different patterns of these inhibitory molecules, NG2, proteoglycan, Neurocan, in this case also tenexin C, a glycoprotein that binds to these molecules as inhibitory. And 8 days post-injury, you've got invasion of leptomeningeal fibroblasts and Schwann cells. We actually now know pericytes are invading the center of the injury and forming that fibrotic center we often see in transection injuries and also in contusion injuries and spinal cord injury. So we have, in this case, leptomeningeal fibroblasts expressing NG2 invading the center of the injury. So these are cells from outside the nervous system forming a fibrotic center of the scar. Neurocan is a central nervous system specific proteoglycan, so it's found in injury margins. Tenacin C is a glycoprotein that will bind to both of these molecules, NG2 and neurocan, so it's found in the center and margins of injuries. And the take-home message is that any axons trying to cross the injury site are faced with multiple axon growth inhibitor molecules in multiple boundaries, so they're wanting to have trouble crossing the injury. So we started wondering, okay, who's making the inhibitors? And so we went to really high power with the concave, we cranked it up, and we used different markers. And the idea was that it's astrocytes that can form scar tissue, as well as I showed you earlier, being supportive of axon growth away from the injury. Astrocytes that form scar, and they'll make neurocan. And so we looked at high power and found that in studying for astrocytes with GFAP, glial-fibrillary protein in red, there's a lot of astrocytes in the margin of the injury which are not expressing neurocan in green. So we went to really high power, and we found in fact that it was NG2 positive glial precursors, a type of stem cell that can make glia. Remember, the glia, that's the general term for astrocytes and oligodendrocytes. That's the Greek word for glue. It shows you how important people thought they were in the past. I'll show you how important astrocytes are in a minute. The take-home message here is that, in fact, that we found that there were endogenous stem cells, glial precursors, that were contributing to scar formation in these injury sites, expressing molecules like neurocan and NG2 on their cell surface. And so this gave us the idea in these early studies in 2003, hang on a second, you might have to be careful how you use stem cell technologies to repair central nervous system. If the nervous system is in part using its own stem cells to try and form scar tissue and contributing to that inhibitory matrix that you find at the injury site. So this gives the idea early on, be careful. If you put more naive stem cells in, maybe they'll also get turned into scar tissue. So, cell-based therapies for spinal cord injury. Background, as I talk about the actual stem cell experiments. So, what is the best candidate? What are the best cell types? At the moment, there's a lot of focus on taking cells from other parts of the body and transplanting them into the injured nervous system. We've got Schwann cells from peripheral nervous system, because peripheral nervous system regenerates reasonably well compared to the injured spinal cord. People looking at engineering fibroblasts to release growth factors. There's a lot of interest in taking olfactory gene cells from the nose. Once again, cell types have been shown to be supportive of axon growth, and a variety of other cells, marrow cells, umbilical cord cells, mesenchymal stem cells as well, a whole variety of them. These are taking cells from other parts of the body, and the general idea is that in many cases, these are relatively easy to get hold of compared to trying to get hold of astrocytes, certainly primary astrocytes. You can't take them out of the patient. And so this is what's been really driving use of these technologies. And I would say in some ways, it's almost like taking parts from your refrigerator, trying to repair your car. Both mechanical, it's not the best match. Shouldn't we be thinking about using central nervous system cells to repair the central nervous system? And so people have tried using immature astrocytes in the past, but it didn't work very well. Immature astrocytes didn't work very well. People have tried using neural stem cells, and they're making a hullabaloo in the press. But when you look at the actual data, you're getting 10–15% effects, which may hit statistical significance in preclinical studies. But I would argue we have to have really robust effects in preclinical models to really warrant translating these technologies to use in humans. People have tried also glial progenitors, and I'll explain a bit more about what they are in a second, a type of stem cell. And they've had some sort of success, but I think we can do a lot better. So, best candidate cell types. Cells of CNS origin. We've seen in these old studies that adult astrocytes away from the spinal cord injury were supporting axon growth, and yet there were also astrocytes forming glial scar tissue and contributing with the stem cells to a formation of this inhibitory environment. And it's really embarrassing, actually, as a neuroscientist to stand in front of you and say how little we know about different types of astrocyte in the central nervous system. And yet they're one of the most abundant cell types. I mean, some studies say that in the human nervous system in particular, as many as 50% of central nervous system cells can be characterized as some kind of astrocyte. And neurons may only make up 10–15% of the central nervous system. So it's really, as I say, embarrassing how little we know about different types of astrocyte in normal nervous system, let alone what types of astrocyte you might make from a stem cell. So what do we know about astrocytes in general, however, that would make them good candidate cells for repair of central nervous system? So the old idea is that, well, astrocytes provide metabolic support for neurons. You can see an astrocyte here with end feet on a blood vessel. They help form the blood spinal cord barrier, and they are very important for metabolic support of neurons, undoubtedly. These are the old ideas about astrocytes. If you make the right kind of astrocyte, I'll show in a second, you can maybe form a bridge across the spinal cord injury. They're also now known to be important for regulating the ability of glial precursors in the types of stem cells in the nervous system that can make oligodendrocytes, the myelinating cell of the nervous system. And not only can they regulate that remyelination in terms of differentiation of glial precursors to make oligodendrocytes, but they're also important for regulating survival of oligodendrocytes as well. Now there's an emerging interest in how the ability of astrocytes to regulate synaptogenesis and also growth of dendrites. So some great work has been done by Ben Barris's lab at Stanford that's shown that there are factors such as thrombospondins released by astrocytes that actually promote synaptogenesis, formation of synapses between neurons. BDNF is a factor released by astrocytes that can also promote neuronal dendritic growth as well. And so now we're realizing that astrocytes have processes which sit at synapses, that synaptic cleft. And not only do we have neurotransmitters now, but we have gliotransmitters in terms of astrocytes can take out neurotransmitters from synaptic cleft and actually regulate activity of synapses by influencing with their own gliotransmitters. And so the idea is that one astrocyte in the human cerebral cortex can regulate the activity with activity of 100,000 synapses around it. And we now also know that the astrocytes have their own signaling networks, calcium waves propagated through astrocytes. So now we're understanding the chemical signaling between networks of astrocytes that runs in parallel to the action potentials we see running through axons in the central nervous system. We discovered neuronal networks and axonal signaling about 100 years or so ago, thereabouts. And so we got into our idea that the way the nervous system wired is just like the wires in our house. To a certain extent it is, but we were ignoring the signaling that takes place in these support, which is the glue support cells, the astrocytes. We're now realizing that they are just as important as neurons and may actually regulate a lot of the activity of neurons in terms of signal propagation, whether they make synapses or not. So I'd argue that we realize that astrocytes are essential components of brain and spinal cord neural networks. So can we make the right kinds of astrocytes for spinal cord injury repair? So of course we turn our attention to stem cell technologies. And the embryonic stem cells are termed pluripotent. The idea is they can make any cell in the body. But we all know in fact that they don't directly make different cell types and tissue. They go through a series of their more restricted stem cells that are more restricted in terms of what types of cells they can make in different tissues. And there are stem cells that just make liver, or make bone, and there's more restriction of stem cells before we actually end up with the final mature cell types of that particular tissue. So in considering nervous system, we know that there are neuroepithelial stem cells that just make nervous system. And then further downstream, we have neuron-restricted precursors. So these are multipotent stem cells, we call them precursors, that will just make neurons. There's a little bit of controversy about these theories, but I think now with the lineage, clonal lineage studies that are taking place, we're realizing that there are distinct populations of cells that will just make neurons, or in this case, just make glial-restricted precursors. So we've been working with Margot Meyer-Brichel at the University of Rochester, who first discovered the earliest types of stem cells in the nervous system, glial-restricted precursors, that are restricted to making just glia and won't make neurons. And in adult rat spinal cord, embryonic spinal cord, it's E13.5 is where these cells can be purified from the embryonic brain and spinal cord, and they're highly proliferative and you grow large numbers of them in tissue culture. So it's these cells we became very interested in terms of making astrocytes as a type of glia. So we knew there were two classic, those are three pathways. You can make oligodendrocytes rather from these glial precursors, and we also knew that there were two classic astrogenesis signaling pathways. One signaling pathway would be BMPs, BMP2 and 4, bone morphogenic proteins, and the other would be neurotrophic factor, or other molecules that hit the GP130 receptor on these glial precursors. And by taking sets of glial precursors, purified glial precursors in tissue culture, and giving them either BMP or CNTF in separate tissue cultures, we can now make separate, defined subtypes of astrocytes in tissue culture, large numbers of them. Pure populations in rapidly, in just 7 days. So we could make what we call our GDase BMP, that's a glial-restricted precursor derived astrocyte by the BMP pathway, or GDase CNTF by using CNTF to induce the precursor to make these types of astrocyte. And they look very different. And they also have very different characteristics in terms of their expression of proteoglycans, the CSPGs, the inhibitory molecules I told you about a minute ago. Turns out that these GDAs BMP have low levels of CSPGs and a high supporter of axon growth. And these other type of embryonic astrocyte, the GDA CNTF, we can make from an embryonic stem cell, look somewhat like the types of astrocytes you find in glial scar, which have high levels of CSPGs and are relatively poorly supportive of axon growth. And we've made these two different types of astrocyte from the same embryonic stem cell, precursor. And now the question would be as well, because we're going to be focusing on in particular on the GDAs BMP as being a beneficial cell type, what different types of stem cells could you use to make beneficial types of astrocytes? And we can talk about more of this later if we have time for questions. And the idea would be, okay, you can use embryonic stem cells, or you can use fetal stem cells. You can extract glial precursors from fetal tissue, which I'll show you in a minute, human fetal tissue. Or the idea would be to now use the new inducible pluripotent stem cell technologies, IPS technologies. But the big question there is, how STEMmy is your stem cell? Are these IPS cells really capable? Are they pluripotent? Can they make all different cell types you can find in the central nervous system? Because we'll be hitting that question later. And be very careful what types of stem cell you ask to make a particular type of astrocyte as well. Okay. So we're going to focus on the GDAs BMP, put them back into our preclinical spinal cord injury models, and now test the ability of these cells to make bridges and promote functional recovery in terms of location in the animals. So the first thing we looked at was the ability for these transplanted astrocytes to make bridges. We went back to our adult rat spinal cord injury model, make a discrete injury in the dorsal columns, and now the idea is we're going to inject our GDAs, we put in different types, the GDAs BMP, the GDAs CNTF, or we could take the stem cells that we used to make those cells and put them in instead, and test the ability of these three different cell types to make bridges across spinal cord injuries. Inject them in. And ability, with BDA tracer, we've got the ability of now of endogenous axons, cut axons at the injury site to press scissors to spinal cord. Can they take advantage of an injury bridged for these different cell types? So the bridge is in red. This is our GDAs BMP, our beneficial type of astrocyte, filling the injury site. You see outlined with the white lines here, the injury site. And this is one 20 micron thin histological section aligned with the trajectory of the tract. And we're looking at the ability of axons to grow from this side across here. And you're catching snippets of axons because they're going in and out of the planar section of the 20 micron thin histological section. So if we actually, quantitative analysis, serial section analysis to try and quantify how many axons are getting across the injury site. And we look at our labeling efficiency, and we have 100 axons, let's say percent labeled before the injury, we have a certain pool that are labeled, and say, okay, that's 100% of how many axons we have labeled. We saw 60% of those axons were able to make the center of the injury, and 39% on average made it 1.5 millimeters beyond the injury, and some actually grew up towards reached the dorsal column nuclei. And that all happened in 8 days. In fact, the previous record for axon growth across a similar transection injury in rats with mesenchymal stem cells, growth factors, cyclic AMP in the kitchen sink in there, they actually got 5% axon growth to 1 to 3 months. So we're getting an efficient bridging of the injury site with these astrocytes. We put a central nervous system cell into the CNS. It's a good match. If we take the stem cells that we used to make the astrocytes and put them in instead, and you can see in red, they don't make a good bridge. You see the axons are piling up at the injury margin, piling up here, and there's very few, if any, axons getting across the injury site. So we went to high power and looked to see, okay, what did our naive stem cells we put in instead? What did they turn into? And like a lot of scientists that are transplanting neural stem cells into the nervous system, our stem cells turn into astrocytes. But there seemed to be a type of astrocyte that wasn't supporting axon growth. And you can see that there's co-localization of our marker for our Lyosha precursors, human placental alkaline phosphatase, and GFAP, the making a lot of astrocytes here. And the axons are piling up in this injury margin, forming dystrophic endings. So putting in naive stem cells and hoping that somehow the injured tissue and the stem cells would know how to interact in this case wasn't a good idea. We didn't make a bridge, if you want to ask these cells to make a bridge, despite the fact they made astrocytes. If we take that other type of astrocyte we can make from the embryonic stem cell, the GDA CNTF, in general, the idea is, well, young embryonic cells are good for repair. Put them in instead, And you can see this GDAs, another the astrocyte in this case, made in tissue culture, GDA-CNTF, the axons won't cross. Compared to another set of animals where we put the right type of astrocyte in GDAs-BMP that we were making tissue culture, and we get good axon growth across the injury. So if we actually compared, control would be make an injury, no bridge at all, we get 4% of axons crossing the injury. Any lab that tells you this was 0 crossing, I'm sorry, Cal showed us 100 years ago that some axons that get around an injury. And so it's 4% as well. So I took a lot of flack actually for putting that data out there, but it's real data. So put in our stem cells, our Lyosha Precursors, next to no difference, statistically not better than doing nothing at all. Put in our GDA-CNTF, once again, we're getting a trending now that's statistically not better, and of course 60 and 39%, which is the middle of the injury and beyond, if you put the GDAs-BMP in. So what about functional recovery? It's already very well making bridges. Did the rats walk better? So we then switched to a different injury model, one that was better suited for really analysing functional recovery. And we made, once again, discrete injuries, transection injuries in this case, and we're now doing contusion, but these are transection injuries in these studies, of the dorsolateral funiculus white matter. In particular, we're hitting the rubrospinal tract, which I know is not so important in humans, but in rats it's really important for targeted paw placement during locomotion. So we're hitting this, cutting this pathway, and remember the neurons in the brain don't like having their axons cut, so we're looking at red nucleus inhibitor too as well. And so we can also then use what's called the grid walk analysis or horizontal ladder to test the targeting ability of these rats to place their paws on rungs and see if we get a good functional recovery or not. And so in the next graph I'm about to show you, down is good, up is bad, in terms of numbers of mistakes being made by these rats as they cross the ladder. So prior to injury, these rats are making 1 to 2 falls per run. After injury, they're making between 5 and 6 falls per run. It's a nice discrete injury. It's a rigorous test of paw placement with nice tight error bars, 8 to 9 animals per group. And those animals that receive the Lyosha Precursors or no treatment at all are making 5 to 6 falls per run, and we take them out actually for many months and they never recover, which you'll see in a minute. Those animals that receive an acute spinal cord injury, the injection of our GDAs-BMP right into the injury site, by 3 days they're making half the number of mistakes. By 14 days, they're not statistically different from pre-injury scores. It's a discrete injury, but it's a remarkable result. Okay? And the stem cells here gave us no benefit at all. If we could take that other type of astrocyte and put them in instead, in the red trace here, we see that they're not giving us any recovery either, the GDA-CNTF. Here's another set of animals. Transplant survival wasn't quite as good, but we still got good functional recovery in these animals. It's real data. It's published in Journal of Biology. So I mentioned that if you cut the axons, they don't like having supraspinal axons. The neurons don't like having their axons cut. So you look in the red nucleus, it's been well characterized by the labs that 50% of neurons in the red nucleus will undergo atrophy, particularly in the magnocellular region of the red nucleus, the long-distance projecting neurons. And so we went and did an analysis of neuron atrophy with and without GDAs-BMP and the other cell types. So before injury, nice big fat healthy neurons in the red nucleus. After injury, a lot of atrophy. Put GDAs-BMP into the spinal cord and wait a few weeks later and look in the red nucleus, you've got a robust neuroprotection of these neurons in terms of suppressing atrophy. So control 50% of the neurons have atrophied. The Lyosha Precursors into the spinal cord, no benefit. Same with the other type of astrocyte, GDAs-CNTF. Those receive GDAs-BMP. Only 18% atrophy, a robust increase in neuron survival. This makes us think about the applications of these cells in a number of neurological disorders. So now we'll quickly talk about, in the interest of time, a dirty secret with Stemcell Technologies thinking about repair of spinal cord injury in particular. This was a seminal study from Lars Olsen's group that came out in 2005 in Nature Neuroscience. And what he's showing here is, in fact, as this title suggests, that there were problems with allodynia, pain syndromes, developing in rats that were transplanted with naive human neural stem cells. And that he then was trying to pre-differentiate them into oligodendrocytes. He saw that with the benefit he was trying to make astrocytes. But one thing that happened was he saw that the majority of his neural stem cells turned to astrocytes, and this correlated with induction of pain. And there's a great deal of interest in the pain field in the role of astrocytes in promoting pain syndromes after various traumas and insults to the nervous system. So we knew that our neural stem cells, had to make glial precursors before they could make astrocytes. So we were really rather worried about this because it's all very well having a cell type that may promote a good axon regeneration and functional recovery, all these pain syndromes as well, then it's obviously not something that we want really to people. So we run a few labs that actually will test for induction of pain in animals. And so there's a lot of work here, and I'm not going to go into all the details in the interest of time. But the bottom line is here that down is bad, up is good in terms of induction of mechanical allodynia with von Frey filaments, testing rats, different time points, weeks, and different colours here, weeks after injury and transplantation of cells into the injury site. And we're seeing that the GDS-CNTF and the glial precursors are both inducing pretty robust mechanical allodynia and thermal hyperalgesia as well, tested with the hot plate test. And what's interesting is it takes a week longer for the glial precursors, the animals treated with these cells, to start showing these pain syndromes. The idea might be that these cells transplanted in are now being differentiated to maybe a GDA-CNTF type astrocyte, because we know that CNTF in the injured spinal cord, and maybe they're driving these other precursors to types of astrocyte that are not beneficial. Lucky for us, the GDA's BMP did not induce mechanical allodynia or thermal hyperalgesia. We actually followed this up by actually looking at sprouting C fibers into the spinal cord in these animals treated with these different cell types. And quickly, basically, we're seeing a doubling in the number of C fibers sprouting to lamina 3. They're expressing calcitonin gene-related peptide, CGRP, sprouting to lamina 3, doubling in the stem cell treated animals, and those who receive GDS-CNTF. We did not see that effect. In fact, it's a trend towards suppression, sprouting the animals that receive the GDS-BMP, the beneficial astrocytes. GDS-BMP, no pain or gain. A little flippant, but yeah. Okay, so that was rat studies. We just published last year, I think it's all right saying it's the first study. We've now taken human fetal glial precursors from E65 spinal cord, and we can isolate those cells, we can fact sort them out. They have many characteristics of the rat glial precursors, the stem cells, and we can drive them, these cells once again with BMP or CNTF into two different types of astrocyte that have very similar characteristics to the rodent cells. So we knew our spinal cord glial precursors, we knew the difference in terms of expression of GFAP, a lot more GFAP than GDS-CNTF. Proteoglycans are up, more so in the CNTF, NG2 is up. That's for rodents. And the human cells came out exactly the same way. More GFAP in the human GDA-CNTF, more phosphocan, and more brevican in the British proteoglycan. We also looked at other characteristics that were important for neuroprotection, and particularly glutamate transporter 1 was up in a beneficial human astrocytes, GDS-BMP, and another interesting result was glial-derived neurotrophic factor, majorly up in our human GDS-BMP compared to CNTF's stem cells. So same model, root spinal tract injury model, transplant now human glial precursors or the two different types of astrocyte. First thing we looked at was the ability to support axon growth. These are cross sections of the injury site filled in red with either human GDS-BMP or human GDS-CNTF, and at the end on in green are axons. And you can see there's a far higher density of axons that are growing into and actually also found out of the human GDS-BMP grafts compared to the human GDS-CNTF. So we're getting that same biology now found in the human stem cells and the human astrocytes we can make as we saw for the rodent cells. So then we actually did what I consider somewhat of a crazy and somewhat heroic study, in that we actually looked at survival of different types of neuron, different lamina, the spinal cord adjacent to these transection injuries that were treated with different types of human cells, two different types of astrocyte or the human glial precursors. And we're doing using UN analysis, we're looking at different lamina, 9 where we find a lot of motor neurons, 7 we have motor neurons and interneurons, and these are the lamina here we have a lot of interneurons, which in particular in the rat called the spinal tract innervates, which will then also innervate neurons. So there's a lot of data here, and I'll quickly go through this. The idea is that we're looking at many rats, and we're looking at serial section analysis above and below the injury and adjacent to the injury, and looking at compared to no treatment at all, which would be 0, how much better off are we in terms of survival of neurons in different lamina and spinal cord. And what we're finding here is that there were two sets of animals that received two sets of human GDase BMP, and we're seeing robust 40% increase in overall numbers of neurons. That would be through 4 to 9, all the lamina and spinal cord. Motor neurons in particular, we're seeing robust recoveries. We're seeing some effects for these GDase CNTF, particularly motor neurons. In fact, if you go to look right immediately adjacent to the injury site, the GDase BMP are giving us a 70% increase in survival of motor neurons in the spinal cord below the injury site. The glial precursors alone are giving us, what was it, about 30-odd%. There's a clinical trial right now for ALS, where they're transplanting human glial precursors to promote survival of motor neurons below the injury site. And we agree with them, yes, you'll get a 30% effect. But if you differentiate those glial precursors to our GDase BMP, you might have achieved 70%. And look at the other lamina. Lamina 7, and particularly 4, 5, 6, there are trends towards deficits in numbers of neurons in those animals that received the stem cells or the GDase CNTF. So it's not enough just to get good survival of your motor neurons. What about the other lamina? All those circuits are going to work together to give good spinal cord function. We also looked at the ability of these human cells to promote functional recovery. And as you'll see here, down is good, up is bad, same model, horizontal ladder. The GDase BMP are giving us good functional recovery in the rats, and the GDase CNTF, human GDase CNTF are not. And we also compared another study, human glial precursors, once again gives no benefit compared to no treatment rather, and human GDase BMP gave us recovery again. We had problems with cell survival because we used methylprednisolone instead of cyclosporin A, and it wasn't as good, but it's real data, it's still came out significant. This was the first time that two different types of astrocyte made from human stem cells could be shown to have different effects on the pair of central nervous system. So, are all stem cells competent to make human glial precursors competent to make the right kinds of astrocytes when you give them BMP? So in this study, we actually were able to use human embryonic day 105 spinal cord, extract human glial precursors. Remember, the original study was E65. Now we're going to 105. You get out glial precursors. Science tells us these stem cells are the same. They get the same name, the human glial precursors, even though they're from older tissue. Give them BMP or CNTF, make astrocytes, test them in rats. No functional recovery at all. So it's not enough to give BMP to a human glial precursor and make an astrocyte. You've got to have the right age of glial precursor, developmentally right age. And we know now there's a change in the BMP receptors in development from younger versus older glial precursors. So now we're going to have to get a lot more sophisticated about how we use stem cells in terms of not only putting in naive stem cells, that's not enough, and hoping for the best that they're repaired tissue. We get lots of nasty side effects and no recovery. But even now, be more sophisticated about what age of glial precursor we treat with BMP, and be careful what type of astrocytes are made. So I'm going to finish up now by talking about chronic spinal cord injury. Those were all acute studies. And so we see here this transection injury where we've waited 9 months after injury. And look at the nasty astrogliosis and change we're getting in the white matter beyond the injury site. It's almost like elements of the scar are now found beyond the injury site in these long-term chronic animals. We're one of the few labs that will go this far out in our preclinical studies. So can you imagine if you made a bridge here with any cell type? You make this more permissive, but now you're running into more problems further down the track. And so little is understood in human patients because not many have transection injuries, but the understanding is from talking to people like Byron Kakulis, who has looked at a lot of human spinal cords as a pathologist, so we have these astrogliotic changes in human spinal cord injury as well. Okay. So, what can GDase BMP do to promote functional recovery in chronic SCI? Well, we thought making a bridge wouldn't necessarily be the best way of using these cells because of these problems in terms of tract ability to support axon growth beyond the injury site. So we took another approach. We knew that astrocytes were important for forming, in terms of for promoting plasticity, connection, and synaptogenesis. So we transplanted our cells just beyond the injury site, so below the injury site, and a dorsal atrophy injury. and now look to see whether these cells could promote plasticity and look to see if it could give functional recovery when used in this way. So the idea is we're now transplanting these cells into grey matter adjacent to motor neuron pools, and now look to see below the intracyte whether this will also be of benefit. So we have good transplant survival, GDS-PMP versus Lyosha Precursors, put into grey matter below the injury. And we're interested in looking at changes in this molecule synapsin-1. It's a presynaptic vesicle protein, which is really a good indicator of synaptic activity. Oh, by the way, of course, these were transplanting cells 1 month after injury, these animals, and a full 8 months after injury as well, long-term chronic spinal injury. Okay. And so we're also interested particularly in what's happening in the motor neuron pools. Obviously, in changes in synaptic activity. And so looking at the changes in synapsin-1, we find that those animals that receive the GDS-BMP compared to control, if you have a 67% increase in synapsin-1 in the motor neuron pools, in animals that were treated 1 month after spinal cord injury with the astrocytes. Compared to the glue precursors, you're actually 95% better. It's two-way and over analysis that's all statistically significant. Okay. So what about functional recovery? So once again, it's the horizontal ladder model. And what we're finding is we treat these animals at 1 month after injury, when they've stabilized in terms of the number of mistakes they're making. And at this point then, we find that the GDS-BMP animals, it takes a while to get going. It takes 2-3 weeks to really get going. And now there's a significant reduction in numbers of mistakes being made by these animals compared to those who received the stem cells or had no treatment at all. So we've also gone out 8 months after injury with this dorsal atrophy syndrome model and get nice stable deficits in the animals at 8 months. So at 8 months after injury, we now transplant these animals with the GDS-BMP versus the stem cells, the glue precursors. Once again, you don't see anything for the first 7 days or so. And then at 3 weeks, we start hitting statistical significance. Once again, this is a long-term chronic spinal cord injury in rats. We're now moving to contusion injury models as well, and it's looking very promising. I'm just about to finish here. So in summary, controlled differentiation of your stem cells, in this case Lyosha Precursors, I think is required for optimal repair of the injured nervous system. There's a lot of money being put into stem cell technologies. And I think we're at the very early stages of using these stem cell technologies. There's a lot of money being put into taking stem cell and putting it into injured tissue and hoping for the best. And I think that we can get some benefit from that, but we can do far better if we control what stem cells turn into in terms of cell replacement, rather than just using stem cells as to release growth factors. You might use a pump to do that. Not all human glue precursors are competent to make beneficial astrocytes. Be careful what type of stem cell you ask to make an astrocyte. Not all cells you can make from stem cells are necessarily good for repair. In fact, you've got nasty side effects, such as pain syndromes when you put the wrong kind of astrocyte in, or left the stem cells to their own devices in the injured nervous system, and it was didn't get functional recovery and had pain syndromes. Transplantation of specific types of astrocyte to acute chronic spinal cord injury can promote repair. Now we need to scale up these technologies and find out multiple crafts, how does that work with neurosurgeons as to how we can translate these technologies. And I hope I've convinced you that in terms of stem cell technologies for repair of the nervous system, we really should be looking at the glue, the glia, the astrocytes in particular, and these should be now a major focus of stem cell technologies. I'd like to acknowledge so many people who conducted these studies. Davies, who's my spouse and colleague, is the person who actually works in the lab, and she's an assistant professor, and really helps drive these technologies in terms of developing techniques and means of differentiating cells in culture. Suzanne Green did a lot of behavior studies. Christophe Brichel is our collaborator at the University of Rochester, who really helped particularly with the deriving the human glial precursors, extracting them and making human astrocytes. Thank you very much.